Hello everyone and welcome. My name is Alfred. So this is our fourth presentation of our ongoing immunology series, and in this presentation we are going to discuss about immunoglobulin genes. We shall also discuss the concept of immunoglobulin class switching, hybridoma cells, and how hybridoma cells are used for production of monoclonal antibodies. So first thing, any substance can elicit an immune response, be it pathogens, synthetic proteins, or any molecule that can interact with our immune cells. And there exists a huge repertoire of B cells, that is about 1 to 100 million clones, which exist with unique surface proteins. And the humoral immune system is capable of producing approximately 100 million structurally distinct antibodies, and each of the structurally distinct antibody has a unique amino acid sequence. Therefore, each antibody is encoded by a different gene. So if one gene and one antibody hypothesis was correct, then we would have required billions of genes to produce billions of structurally distinct antibodies. But you needed to know there exists a fine number of protein encoding genes in our genome, which is approximately 20,000 to 25,000, and out of these only few hundred genes are involved in producing antibody. So the question now is, how is small amount of genes producing such diversity of antibodies? And in the course of this presentation we are going to answer this question. So next we have to look at the location of immunoglobulin genes, and for each type of antibody chain, that is for the kappa and the lambda light chains and the heavy chains, they have a separate pool of gene segments located on different chromosomes. So each pool contains a large number of gene segments encoding the V region of an antibody chain and a smaller number of exons encoding the C region. And here we can see the arrangement of the V gene segment, the J gene segment, and the C gene segment, and even the D gene segment on different chromosomes. So for the kappa and the lambda light chain, this V gene segment, J gene segment, and the C gene segment are going to be located on chromosome number 2 and chromosome number 22, that is for the kappa and the lambda respectively. Whereas for the heavy chain, the V gene segment, D gene segment, J gene segment, and C gene segments are going to be located on chromosome number 14. So you can see for the heavy chain, we are having an additional D gene segment found between the V gene segment and the J gene segment. So V stands for variable region, J for the joint region, C for the constant region, and D for the diverse region. So a pool of gene segment will contain the V gene segment, J gene segment, and C gene segment, like I've already pointed out. And in the case of the heavy chain, we have an additional D gene segment that is located between V and J gene segment. So for the kappa light chain, chromosome number 2 have 40 V gene segments, 5 J gene segments, and 1 C gene segment. And for the lambda light chain on chromosome number 22, there are about 30 V gene segments, 4 J gene segments, and 7 C gene segments. Then for the heavy chains on chromosome number 14, there are about 60 V gene segments, 27 D gene segments, 6 J gene segments, and 11 C gene segments. And you need to know that the V gene segments, D gene segments, J gene segments, and C gene segments are not continuous. So they are widely separated from each other. So what are the benefits of gene rearrangement? So gene rearrangement number 1 brings up together the separate gene segments and C region axons of the antibody gene, and they also activate transcription from one gene promoter through changes in the relative positions of the enhancers and silencers acting on the promoter. So therefore a complete antibody chain can be synthesized only after the DNA has been rearranged. So next we move on and discuss mechanisms of antibody diversity. So there are several mechanisms of antibody diversity. These include multiple germline immunoglobulin gene, somatic recombination, junctional diversity, somatic hypermutation, and multiple combination of light and heavy chains. So we are going to discuss this one by one. So starting with multiple germline immunoglobulin gene, we have said that on chromosome number 2, 22, and 14, we have multiple gene segments that are involved in encoding of immunoglobulins. So the large number of inherited V, J, and D gene segments available for encoding antibody chains make a substantial contribution to antibody diversity. Moving on to somatic recombination. So before we discuss somatic recombination, it is important for us to first of all have an overview of B cell development, or the origin of B cells. So during embryogenesis, the precursor cell of B cell are first recognized in the fetal liver and then migrate to the bone marrow. So in adult life, bone marrow is the site of B cell development. So in the bone marrow, the stem cells are going to differentiate into the pre-B cells. And pre-B cells can express mu heavy chains. That's why you can see in this diagram I have indicated mu here inside the pre-B cell. So pre-B cell express mu. And in our previous presentation, we know that mu is an example of a heavy chain. So you need to take note of that. So we are saying pre-B cell is going to express mu, heavy chain. So however, since we need both heavy and light chains to make an antibody, pre-B cells cannot make any antibody because they lack the light chains. So what is going to happen is that the pre-B cells are going to be differentiated into the B cells, and B cells have the capability to make both the light and the heavy chains. So they will combine the light and the heavy chains to make an antibody. And you need to know that the first antibody that will be produced is immunoglobulin M, and they will go to the surface of the B cell. So you need to know that from B cell, from the stem cell, that is, to B cell development, the process is antigen independent. So you have to take note of this. So from the stem cell all the way up to the B cell, the process is antigen independent. Very important for you to know that. So what is going to happen next is that the activated B cells will differentiate into the plasma cells, and we also know that the plasma cells have the capability of secreting antibodies. So after looking at B cell development, so now, how does somatic recombination bring about antigen diversity? So somatic recombination occurs during B cell development, and during somatic recombination, specific combination of single V and J gene segments is selected for the light chain. Similarly, a specific combination of single V, D, and J gene segment is selected for the heavy chains. So the DNA segment that were separating these selected single V, D, and J gene segments are going to be deleted. And what is going to help us in deletion is an enzyme called enzyme recombinase. So the deletion is carried out by an enzyme recombinase that is encoded by the RA gene 1 and RA gene 2. So RA genes basically represents or stands for recombinase activating genes. Now, there is a question which we have to ask ourselves. All somatic cells have chromosomes number 2, 22, and 14. But why can't they produce antibodies? Or why is it that they don't produce antibodies? So you need to know that only B cells and plasma cells have the capability of producing antibodies, whereas other somatic cells cannot produce antibodies despite the fact that they still have the gene segments that are needed to produce antibodies. And this is because only the B and the T cells can express recombinase with the help of RA gene 1 and RA gene 2. So that is the reason why it is only the B and the T cells that can produce antibodies, is because they are the only ones that can express recombinase enzyme. So recombinase will initiate double-stranded DNA breaks at specific DNA sequences that were flanking the selected V and D gene segment. Then what is going to follow next is that the gene segments that were separating the selected V, D, and J gene segments will be deleted, and the undeleted V, D, and J gene segments will be joined by the help of ligase, then rearranged. So the DNA sequence will be transcribed into an mRNA that contains the codes to make immunoglobulin light or immunoglobulin heavy chains. Now, what happens during DNA rearrangement? So this is a very important thing that you need to understand, and we are going to explain it using the next diagram before we come to this one. So we'll use this diagram to explain what happens during a DNA rearrangement. So during DNA rearrangement, randomly selected V gene segments will be translocated to lie close to a single D gene segment, several J gene segments, and a single C gene segment. So as we can see from this diagram here, we are having several of the V gene segment from the germline heavy chain. So one of it is going to be selected. For example, for this case, they are going to select two. So they will select the second one. So that is what is going to be selected. So that one V gene segment will be selected, then lie close to a single D gene segment. So on this part we have the D gene segment. So you can see the D gene segment. So one of it will be selected, and that is, for this case, we have selected B. So the V gene segment will lie close to a single D gene segment and several J gene segment. So the J gene segments are here. So we have the J gene segments from 1 all the way to 6. So they will lie close to several J gene segments and a single C gene segment. So for our C gene segment here, it is mu, so it will lie next to a single C gene segment, which is mu for this case. Now, what is going to follow next? So this is followed by transcription of the heavy chains. So the rearranged DNA, a heavy chain, is going to be transcribed. So it is going to be followed by transcription. So you can see these rearranged DNA segment of the heavy chain will undergo transcription. And you need to understand that during transcription, RNA is produced, specifically the mRNA. So mRNA is going to be produced. And in the mRNA, as we can see, there is one V gene segment. So we have one V gene segment, this one V gene segment that you can see there. One D gene segment, the one appearing in black. We have one D gene segment. But we have several J gene segments. So you can see the J gene segments are several. So we have several of the J gene segment running all the way from 2 to 6, 2, 3, 4, 5, and 6. So we have several of the J gene segment. But you need to know that only one J gene segment is needed. We only need one J gene segment. Now, how are the other J gene segments going to be removed? So the other J gene segments are removed by the help of RNA splicing. Very important. That's why you can see here I've indicated RNA splicing. So with the help of RNA splicing, we are going to remove the other J gene segments and only remain with the one J gene segment. So the final RNA encoding the heavy chain is translated to form a heavy chain protein. So after we have splitted the other J gene segment, so the mRNA, sorry for that. So our mRNA is going to undergo translation to give us a what? Heavy chain protein. And since the V gene segment, D gene segment, and J gene segment is lying close to C gene segment, specifically mu, that's why we are saying immunoglobulin M will be the first immunoglobulin to be produced. Very important for you to understand. And the same thing is happening here. So you can see we are having several of the V gene segments. So one of it will be selected randomly, then lie close to a single D gene segment, several J gene segment, and single C gene segment. Then the next thing that is going to be formed, the next thing that's going to follow that is there's going to be transcription of the heavy chain gene. That is, the mRNA will undergo the transcription. And like we have said already, in the mRNA, there are one V gene segment, one D gene segment, but several J gene segments. So you can see the several J gene segments here. So those several J gene segments have to be removed with the help of what? Of RNA splicing. And finally, the final RNA encoding the heavy chain will undergo translation to form a heavy chain protein. So it's very important for us to understand that mechanism. Now, the next thing that we are supposed to discuss is junctional diversity. So during assembly of V, D, and J gene segments, small number of nucleotides may be inserted or deleted at the junctions that are joining these segments, and this will result in a shift in the reading frame that produces an non-functional gene, thus creating diversity. So next we look at somatic hypermutation. So somatic hypermutation is brought about by increased rate of mutation in the V gene segments of immunoglobulin genes. So an enzyme called Activation Induced diaminase, that is abbreviated as AID, is going to induce replacement of cytosine with uracil, and that is going to create a point mutation. So we know that from our basic biology that normally uracil is not found in the DNA. So to preserve the integrity of our DNA, base excision and repair enzymes will come to remove that uracil. And the enzyme that removes the uracil is called uracil DNA glycosylase. So this enzyme will come and try to remove the uracil, which is not supposed to be part of the DNA molecule. Then what is going to follow next is that error-prone DNA polymerases is then recruited. So error-prone DNA polymerases is then recruited to fill out the gaps. But since they are error-prone, so they will make some mistakes, therefore creating what? Creating mutation. So there will be point mutation. And you also need to know that B cells that are making the antibodies with higher affinity for the antigen will be selected to proliferate extremely. And this is what we refer to as clonal selection. So you need to know that B cells invite mutations as they are trying to create more variations in their antibodies. And the variable regions of the immunoglobulin is the one which is involved in antigen binding. That one we know from our previous discussion. So initially, the affinity of the antigen with the antibody is usually low. But as a result of somatic hypermutation, there will be formation of antibodies, some of which will have lesser affinity with the antigen and some will have higher affinity with the antigen. So the B cells that are making the antibodies with the higher affinity for the antigen will be selected to proliferate extremely. And this is what we have said is known as clonal what? Clonal selection. So as a result of clonal selection, B cells that are making antibodies with higher affinity will predominate. So therefore, we end up with the antibodies that will have higher affinity with the antigen. And this concept is also referred to as what? Affinity maturation. So you need to take note of that. And lastly, we are going to discuss concern on a multiple combination of the light and the heavy chain. So during assembly of immunoglobulin, random combination between light chains and heavy chains occur, creating further diversity. So moving on, we are going to discuss immunoglobulin class switching. So we know that during B cell development, many B cells switch from making one class of immunoglobulin to making another. So immunoglobulin class switching is also referred to as isotype switching. And during immunoglobulin class switching, the same assembled variable region gene of the heavy chain is sequentially associated with the different genes in the constant region of the heavy chain. And you need to know that immunoglobulin class switching only occurs in the what? In the heavy chain. So considering switching from immunoglobulin M to immunoglobulin G, we are going to use this diagram to explain how that happens. So we are having the V, D, and J gene segments here. So we can see our V, D, and J gene segments. So the same V, D, and J gene segment is going to be first associated with mu. So we can see this one is first associated with mu gene in the constant region. And that is why immunoglobulin M is the first antibody that our B cell can produce. So suppose we want to associate the same V, D, and J gene with gamma 1, so that we have to put the what? The V, D, and J gene next to what? Next to gamma 1. So we are saying, suppose if we want to associate the same V, D, and J gene segment with gamma 1, so we have to make sure that we have put the V, D, and J gene segment next to what? Next to gamma 1. So how is that going to take place? So the enzyme that is involved in the DNA rearrangement is called switch recombinase. So we will have to rearrange this molecule so that we have V, D, and J gene segment lying next to what? To gamma 1. So what is going to help in that rearrangement is an enzyme called switch recombinase. So the switch regions, the ones that have indicated in black, these are the switch regions. So the switch regions will be activated and they will cause looping out of the intervening DNA. So the switch regions are going to be activated, then they will cause the looping out of the intervening DNA. For example, for this case, we have to make sure that we loop out that portion. So as you can see here, once the switch regions have been activated, then the portion is going to loop out. So you can see the looping out of that portion. Then what is going to follow next is that the looped out portions will be excised and it will be degraded. So in this mechanism, we can switch from immunoglobulin M to immunoglobulin G, A, or even immunoglobulin E. However, different mechanism is involved when we are making immunoglobulin D. So for immunoglobulin D, what happens? So in the case of immunoglobulin D, an mRNA is produced which will contain the same V, D, and J gene segment, and also there will be both mu and delta gene of the constant region. So as a result of that, there will be alternate RNAs slicing, and in that mechanism, B cell will be able to produce both immunoglobulin M and immunoglobulin D simultaneously because of the presence of the mu and the delta gene of the constant region. So basically, that is how immunoglobulin class switching takes place. So next, we are going to discuss factors that can influence immunoglobulin class switching. So we will start with the first one, that is concentration of interleukin. So that is supposed to be interleukin 4 and 5. We are going to use those ones as examples. So interleukin 4, this is supposed to be 4. Please take note of that, not 14. So this one is supposed to be interleukin 4. So interleukin 4 can induce class switching to immunoglobulin E, while interleukin 5 can induce class switching to immunoglobulin A from immunoglobulin M. The next factor is interaction of the CD40 protein that is present on the surface of the B cell with CD40 ligand that is present on the surface of the T lymphocyte. Again, another correction for you to take this, supposed to be T lymphocyte. There is a typo error there. So hyperimmunoglobulin M syndrome is going to occur if such interaction between the CD40 and the CD40 ligand cannot take place. So in that case, no class switching can occur, and in that scenario, only immunoglobulin M will be available. So those are the two factors that influence or affect immunoglobulin class switching. So next we look at the concept of hybridoma cells and how hybridoma cells are used in production of monoclonal antibodies. So hybridoma cells are produced by fusion of different cells. So they have the remarkable capability to produce large amount of single molecular species of immunoglobulin. So in production of monoclonal antibodies, an animal, for example, a mouse, is immunized or injected with an antigen of interest. Then the spleen cells from the mouse is going to be collected and then grown in a culture dish in presence of mouse myeloma cells. Then the spleen chemicals, for example, polyethylene glycol, is used to fuse the two cells. And then what is going to follow next, the culture is going to be transferred to a special culture media called a hut that will promote growth of the fused cells but inhibit the growth of the parental cells. Then ultimately we will end up having large clones of the hybridoma cells, which can then be used to produce large amount of monoclonal antibodies. So basically that is how monoclonal antibodies are produced from hybridoma cells. So this concludes our fourth presentation. I hope someone finds these helpful. So in case you want to know more, you can confirm from your textbooks, and I will also recommend that you comment if you need me to do anything, maybe for your own interest. You can comment, and you can also like my video and share. So until next time, keep safe and be blessed. Thank you.